my name is Ioannis Papantoniou. I am working as a research coordinator at KU Leuven in Belgium. And I have been working over the last 15 years more or less on biomanufacturing, bioprocess development for cell therapies, stem cell therapies and regenerative medicine applications for the last 8 years. In the field, actually, we can say it has gone from a level of 0 to a level of satisfactory. And there is need for customized solutions still, and that's where we are heading to. However, still there were quite some solutions from the biopharmaceutical field already that were readily available from the beginning. Now the issue is how do you redesign and reapply them in the novel biological systems, although they can be like a generic platform to begin with that can satisfy certain needs upstream for different final products. At the end of the day, there possibly will be branching of technologies that are bespoke, that are, customized for the end product. There are a lot of discussions on this topic, right? So do you need to have bespoke solutions all in one? Do you need to have process pipeline? In my opinion, you will need both. So you will need closed automated systems that can execute multiple tasks. The closed nature, okay, it's necessary because we are dealing with products that will be implanted, so you need to have some safety. You need to have also the capacity to monitor online your bioresponses, let's say, and extract information of the process, either if it's linked to your quality or to your potency, or even as a log of your batch. Differences between autologous and allogeneic will have to do with the fact that inherently we are talking about different types of product, one, and two, also we are talking about products that are at different level of maturity. So for the autologous cases, you have right now higher possibilities to reach the patient without too many hurdles regarding regulation or clinical application. Whereas with allogeneic, although they have inherent attractive properties from the biomanufacturing perspective, still it is a high-risk environment for reaching the patient. Now, this creates different biomanufacturing scenario, also regarding the scale, one, and also regarding the fact that you are dealing with, for autologous, donor-derived material. So you have batches that are destined to enter in one patient. So it's patient-specific products, patient-specific bioprocesses at the end of the day. On the other hand, in that case, probably you don't have to deal so much with the storage issue. Maybe yes. On the other hand, you have your large scale, you have your large volumes, economy of scales that might come in, but I think that will be something that will grow in the future and not right now, in my opinion. When you're starting from single cell product, so from a biopsy that you need to extract your MSC population, we are talking of a generic process. You need to expand your cells to reach a certain number that will actually allow the implant to be efficacious upon implantation. So you need to expand your cells. That's a bioreactor process, for example, and you have multiple options there that you need to compare, benchmark with each other for the specific cell type that you are aiming for. So that bioprocess development round, I believe it's pretty generic. So you need to understand donor-to-donor variability, to quantify that. You need to have sensor readouts. You need to make controllers that can actually adapt the process to the needs of the cells that are cultured there. These are generic tools that can be used across the cell therapy field. So that is one part. And the second part is how do you make the tissue in a controlled way? And that's a big challenge because we're talking about three-dimensional structures. I think right now we need to have thoroughly characterized processes. And that means both from the biophysical environment that they create for the cells, one, two, from the sensor readouts that we are getting out of them. So actually the communication we have with our living product, either it's a single cell or a tissue or a cartier, it doesn't matter. And actually, how do we make use of this data in order to be able to make educated decisions about the process? So that's regarding single processes. Then what do we need? We need to have an overarching tool, data management tool, data treatment tool that might unify the pipeline of production. And that is for one manufacturing facility. So if we put this in the globalized context, then again you have a lot of issues to take into account. And again, autologous and allogeneic will have a different scenario of manufacturing. And that's a bit beyond where I would like to be active. It's for other people that are into CDMO, development CMO companies, etc. And from my perspective, it's important, however, to be able to have these bioprocess units that can crosstalk, allowing the whole pipeline to be, in a way, more clever, semi-autonomous, if you can call it like that, and human operations and human decisions complementing it.